So today is our last day in the light reactions. Tomorrow we talk about the Calvin cycle, and then we're gonna kind of all wrap it up together. So the Calvin cycle should only take a day, rather than the light reactions, which is taking 3. So we're just gonna jump right into it because it's a little bit of a longer one today. So light reactions day 3. This one is all about electron flow. So we talked about how the photosystems are set up with the light harvesting center and the reaction center, and how that reaction center is activated when the light harvesting captures electrons, transfers it to a special pair of chlorophyll A molecules, and those special chlorophyll A molecules transfer the electron to an electron carrier. So that's what we did yesterday, and now we're going to talk about how those electrons get from the electron carrier to wherever they need to go. So we're all about electron flow, and there are 2 types of electron flow. So there are 2 different ways, essentially, that these electrons can go through the photosystems, which are in the thylakoid membrane. We are going to be in the thylakoid membrane the entire day. So the first type of electron flow, I'm going to mark with A and B just so I don't get confused with like numbers for steps, is linear electron flow. So this is probably the one that happens the most often, which is why it is the most complicated. It is the flow of electrons through photosystems and other molecular components. All of them that are built into the thylakoid membrane. So I'm going to go through it step by step, writing it out in words, and then we are going to draw it out, because pictures help everything. So step 1 is that the photon is going to hit a pigment in the light harvesting reactions of photosystem 2. So photon hits light harvesting center or complex in photosystem 2. Remember, photosystem 2 happens before photosystem 1. So a photon is going to hit that light harvesting complex in photosystem 2. When the electron falls back to a ground state, because it can't be there forever, that energy is going to be passed to a nearby pigment. So an electron in a near pigment is excited until it reaches the P680 pair of chlorophyll A molecules in the reaction center. So this process is going to happen until it reaches our special chlorophyll A molecules otherwise known as our P680 molecules in the reaction center. Which is what we talked about yesterday. Essentially, this is the process we talked about yesterday. Our electron is going to hit one of those pigments in the light harvesting complex. It's going to excite an electron. When that electron is excited, it can't stay excited for too long, so it's going to drop back down, and that energy will be transferred to another pigment, to another pigment, and to another pigment until it reaches those special pairs in the reaction center complex. Cool? Okay. Step 2. The electron in that P680 is transferred to a primary electron carrier. Now, and when it does transfer that electron, it'll become P680 plus. So when it has that electron, it's just P680. When it doesn't have the electron, it's P680 plus. So that's step 2. Our electron in those special chlorophyll molecules will go to our primary electron carrier. In step 3, an enzyme is going to split water. And it will become 2 electrons, 2 hydrogens, and an oxygen. The electrons will go to P680. The H plus will go to the thylakoid space. I could have written this all out. And that oxygen will go into O2 and be released. So 2 oxygens will find each other, join to O2, and be released. So we've got 1 electron from the primary electron carrier, or in the primary electron carrier. Then these electrons in water are going to bring P680 back, or P680 plus back to P680. Our H plus is going to go into the thylakoid space. This will build up a gradient, just like in protein or in cellular respiration, where we were building up a H plus concentration. 1 is also building up here, and that'll come in handy later. So step 4. Our primary electron carrier in PS2 will go to PS1, or the electrons, in the primary electron carrier. In PS2 will go to photosystem 1 through an electron transport chain. So the electron being carried by the primary electron carrier in photosystem 2 will go to photosystem 1 via an electron transport chain. We've got some carriers in there that we're actually going to name. It's not necessarily like a memorized thing, but just like a kind of be cognizant that this exists kind of thing. They are called plastoquanine, or which is abbreviated PQ. and plastocyanine or cyanin, which is PC. They use redox reactions and pump more H plus into the thylakoid space. Just like the electron transport chain in cell respiration. This first electron transport chain, getting from PS2 to PS1, or photosystem 2 to photosystem 1, will go through these two carriers, and those carriers will carry out redox reactions to pump hydrogen ions into the thylakoid space. Step 5 will be a little bit familiar to you because in step 5, potential energy is used to make ATP via chemiosmosis. Just like ATP synthase existed on the mitochondrial, the super inner mitochondrial membrane, to make or to synthesize ATP with that H plus gradient, the same thing's going to happen right here in the light reactions. We're going to make ATP as a part of the light reactions. So that's 5. While that is happening, light energy is transferred from the light harvesting pigments in PS1 to the reaction center complex. So the same thing that happened in step 1 with PS2 is going to happen now in PS1. So light energy transferred via light harvesting pigments to the photosystem 1 reaction center. Then we have our P700 chlorophyll A's that are now excited. So we do have light being transferred to this primary electron carrier, just like in PS2. So the same system that happened before with the electron bouncing from pigment to pigment to pigment to eventually reaching those special chlorophyll A's. But now that that carrier has the electrons from light, it can now act as an electron acceptor for the electrons from PS2. So, wait, is this still the same step? Yes. So our chlorophyll A's are excited, which will then excite PS1's primary electron carrier, or acceptor, sorry. And now we have P700 to P700 plus, which acts as an electron acceptor for the electrons from PS2. So it's kind of confusing, but we've got our excited electrons that are going to, like from light, that are exciting this primary electron acceptor. And when that primary electron acceptor is excited from the light electrons, it will accept even more, which allows it to be the electron acceptor for the electrons from PS2. So, we doing okay? That was a long step. Okay. Step 7, we're almost there. Our photoexcited electrons are passed via redox reactions from that primary electron acceptor in photosystem 1 down a second electron transport chain through a protein called ferredoxin. So, excited electrons are passed via redox reactions from our primary electron acceptor in PS1 down a second electron transport chain. And in that electron transport chain, we have a protein called ferredoxin, or FD. I'll scroll back up a second. Hold up. Okay. This electron transport chain does not create a gradient. That's why it's important that we differentiate between the proteins in the first electron transport chain and their proteins in the second electron transport chain. Because the ones in the second electron transport chain don't create a gradient. We've got enough of it with the first one. Okay. One last step. In step 8, we have an electron. Or not an electron, goodness gracious. An enzyme. They both started with E. We have an enzyme called NADP plus reductase. Want to take a wild guess as what that does? It reduces NADP plus. So it catalyzes the transfer from ferredoxin to NADP plus to NADPH. And that NADPH is what's going to go to the Calvin cycle. Okay. Now that's a lot. We're going to draw it out and number it, and hopefully that should help a bit. So, why don't you get that down first? Okay. All right. We're going to draw it out. Let's say that this, goodness gracious, this is the thylakoid membrane. Which means this is the stroma, and this is the thylakoid space. Now, instead of drawing like the weird, like corn dog looking thing from yesterday, I'm just going to draw boxes for like ease of space. So you are free to draw this however you'd like, but I'm just going to do boxes. This is going to be photosystem 2. This, I might run out of space, is photosystem 1. So, should I move that? Yeah, so essentially that's a membrane, and this is a protein, and this is a protein that are in the membrane. So just like yesterday when we did so it's like this thing from yesterday, but there are two of them, essentially. I'm going to redraw it just in case. I made it a little bit messy. Yeah, no, so it's just two photosystems. So it's the same thing from yesterday, just twice over, and we're going to talk about the electron transport chains between them. So I'll do circles. Okay. That helps a little bit. So where are we at? Okay. First things first. We have light. Actually, I'm going to keep changing colors because why not? We've got light. So we've got our light harvesting complexes and our reaction centers. So step 1 is that light will hit our pigments. Which will eventually transfer to our two special chlorophylls in the reaction center complex. And that's step 1. Then we have our primary electron acceptor and our chlorophyll A's are going to transfer electrons from the chlorophyll A's to the primary electron acceptor, and that is step 2. You can actually like write these out if you want. I'm going to use abbreviations just for space. Step 3 was the splitting of water. So I'm just gonna come up here. When water splits, we've got electrons, and those will go to those chlorophylls and eventually get transferred to the primary electron acceptor. Step 4 is an electron transport chain where our electrons, now this should be in the membrane. Okay. So step 4 is an electron transport chain. So our electrons from the electron acceptor will go to PQ, will go to a cytochrome complex, which will go to PC, which will eventually transfer electrons to those two special chlorophylls in PS1. And all of this, while this is happening, step 5 is where we get ATP. And we're just doing it like that because I'm not going to draw like a ATP synthase and a proton gradient just yet. But essentially, this electron transport chain right here will be pumping hydrogen ions into this, and then there will be an ATP synthase like over here, and the hydrogen ions will go out the ATP synthase and make ATP, just like they did in cellular respiration. Let's see. Step 6 is light again. I have it coming from this way just for space, but it would be up here. It will hit our little pigments until eventually it gets transferred to the special chlorophyll molecules, which will send those electrons to our primary electron acceptor. Speak again. Step 7 is another electron transport chain where we have our electrons going to ferredoxin. And step 8 is going to NADP plus reductase and giving us NADPH. And that is our end product. So that's linear electron flow. How are we doing? I know it's a lot. So that's linear electron flow. But I also mentioned, we get a 5? Okay, cool. That's linear electron flow. We now have to relatively quickly cover cyclic electron flow. It is not as complicated. So sometimes photosynthetic organisms don't have a PS2. Sometimes they only have a photosystem 1. Or sometimes our electrons just go through cyclic and don't touch photosystem 2. And we'll talk about how that happens. Cool. All right, so we are talking about cyclic electron flow. In certain cases, photoexcited electrons take an alternate path. That uses photosystem 1, but not photosystem 2. That uses photosystem 1, not photosystem 2. In this system, electrons cycle back from ferredoxin to the cytochrome complex, and then to P700. So, yeah, let's see. So electrons cycle back to ferredoxin, or from from ferredoxin to the cytochrome complex. Then to P700. In this process, there is no production of NADPH or oxygen. This is solely used to generate ATP. So this is like if a cell does not need to, or like isn't about to make sugars, they need ATP now. Or if the sugars that they do digest, they don't get from making it, but they do have the ability to like make ATP from sunlight. So if we are drawing it out, we have photosystem 1, we have our light harvesting complex, and we have our reaction center. In our reaction center, we have our 2 chlorophyll A molecules, we have our electron acceptor, and usually those electrons will go to the electron acceptor and then to ferredoxin. Ferredoxin was over here in our last sketch. Ferredoxin will feed them into the cytochrome complex. So an electron transport chain to PC. And then back to our photosystem or or reaction center chlorophylls. So it's just going to go in this circle, and when it does, we will get ATP. So some photosynthetic bacteria only have PS1. They don't have a photosystem 2, so they can't make, or they have to figure out another way to do things. But they can capture light energy and use that light energy as ATP. But because they're usually single-celled organisms, they don't need like long-term sugar storage. So they can just make their ATP directly through photosystem 1. But here's the thing: cyclic flow can happen in organisms with both photosystems. In organisms with both photosystems. We don't really know why. We don't know why this is a thing. We think that it is an evolutionary leftover. Just like we have an appendix, even though we don't use it anymore, we think that photosynthetic cells have this pathway that they can use, but they don't really need it anymore. But both cyclic and linear have the same mechanism for ATP production. Same mechanism for ATP production. Yeah, I think we have time for this. So, how are we doing? Okay. So I don't have an exit ticket for you today, so real quick, I'm going to compare chemiosmosis in chloroplasts and mitochondria, just to kind of loop together what we talked about with cell respiration and what we learned today. So chemiosmosis, chloroplasts versus mitochondria. It's the same basic mechanism. Both use ATP synthase to create ATP by allowing hydrogen ions to flow with their concentration gradient. But the spatial organization slightly differs. That's organization. I realized I used org for organism and organization. We're not going to do that. Spatial organization slightly differs. In mitochondria, I've got symbols here, so let me know if I need to rewrite something. Hydrogen ions flow down their concentration gradient from the inner mitochondrial space. Or inner mitochondrial matrix. Or I guess it's, wait, is it prime or 2? Sorry, I'm going to recheck my notes. Where are we at? Goodness gracious. Okay. Okay, yeah. So they are going to flow into So we are pumping our hydrogens out in cell respiration, right? And we're allowing them to come back into that inner mitochondrial matrix in order to run ATP synthase. In chloroplasts, we've got hydrogen ions flowing from the thylakoid space into the stroma. And that's really the main difference. With our chloroplasts, our hydrogen ions are concentrated on the inside of the membrane, and our mitochondria, our hydrogens are outside of that membrane. Which means our ATP synthase would have to be reversed in its orientation. So if ATP synthase is this way, In mitochondria, it would be this way in chloroplasts. Okay. And we don't have time to draw a giant picture, which is fine. We can do that later. So that is the light reactions. All of that is how we get from a photon of light to NADPH. That NADPH will flow into the Calvin cycle, which we will talk about